I'll start with
Yeah.
first question by Subham. What is reader, writer, and eraser, and how they play their role in genetic modifications to fight with TNBC?
Okay. So as I was trying to explain that the epigenetic landscape of chromatin are fine-tuned by a class of enzymes. So the writer are the enzymes like histone acetyltransferase, histone methyltransferase, which are involved in that importing this modifications into chromatin. Erasers actually remove these marks. That is a histone deacetylase, histone demethylase, like that. And chromatin readers are the protein which physically docks onto these landscapes and are involved in having a multi-protein recognition modules, and they interpret their functions in different biological contexts.
As a follow-up to the question, we have someone asking about what is TNBC?
Okay, so this is triple negative breast cancer. So as I was initially talking about that, there are the estrogen receptor, progesterone receptors onto these breast cancer cells, and that's how these receptors to that chemotherapeutic drug bind. And so in triple negative breast cancer context, this particular receptors are actually missing. So that is why they are negative. So ER, PR, HER2, these three important markers of the breast cancer cells are lacking. That is why they are known as triple negative breast cancer.
Okay, Professor Das, thank you. Moving on to the next question. Do you think the epigenetic regulation of metastasis in the context of breast cancer is mediated by signals in the tumor microenvironment?
Very good question and very relevant and pertinent question in this context. Absolutely, that the tumor microenvironment has huge impact on the maintenance of the tumor cells. And so the epigenetic regulation of the tumor microenvironment harboring cells definitely has an important implication in this, in the context of breast cancer. So actually we are interested and we are working on this. As you have seen that we are working on the benign tumors as well as in the metastatic tumor models. So in the metastatic models, the tumor microenvironment become very important, and so we are actually working on how this microenvironment influences and how epigenetic regulation actually is taking place in this tumor microenvironment.
Thank you, Professor Das. Moving on to the next question. When elucidating such drug mechanisms and doses of drugs, how do you make sure that the off-target side effects are minimal? For example, damage to heart or muscle degeneration, because they can be long-term effect and not immediate effect.
Yeah, so this is a question by Aditi, and yeah, I understand that, and that is very important problem in the chemotherapeutic regimen, because these chemotherapeutic drugs actually lead to lots of side effects. Anyways, the effort from the clinical side is actually to lower down the dosages of these drugs, and so people want to put less drugs or a combination of the drugs so that they actually have less negative effects. But as I was showing in the story, that actually reducing the potential level of this drug actually backfires. It has actually programmed turning on of several other gene expressions which are still bad for the cells. So it's not a linear relationship, that we cannot really scale down the drugs in an attempt to have a control over the off-target effects. So this is something, that is the reason why we have to play around with alternative targeted strategies so that we can have a better response in the context of cancer.
Okay, fine. Uh, Again, there is a question by Aditi. Can you comment on the commercial scalability and cost considerations involved in employing such epigenetic modifications to devise targeted therapies?
Okay, so this is again some interesting part is there here. So so in terms of the translationable uh, paradigm, what we are thinking is, that's why the three-dimensional structure of these chromatin readers, epigenetic regulators, become very important. We intend to design the mimics of these epigenetic regulators, which will have subtle function in the cells, and those particular factors can be treated to the cells like I think you guys know about artificial transcription factors or other things. So like that, if we can devise a small mimic which kind of resembles the function of this particular chromatin reader in the cellular context, would like to treat that to the in the cells and see how that can actually rescue the situation. So that way, we cannot really put in the whole protein in the experimental backdrop. We have to think about how this can be implicated in a cellular context. So structurally, it is a very important problem to get into and understand how we can devise small molecules of these analogs, or analogs of these proteins.
Okay. Thank you, Professor Das. We have a question from anonymous attendee. Do you see difference in the fold change of cancer stem cells marker if you were to use another cell line like MCF7?
Okay. So yeah, so we actually did our studies in all the different luminal lines like MCF7, T47D, as well as triple negative breast cancer models that you have seen, MDB231, MDB468. So there are cancer stem cells in the breast cancer context. The well-known marker is CD44 positive, CD24 negative. That is what we have done in our studies. Looked at CD44 positive, 24 negative cells, and we see that so in this kind of drug treated condition, 44-24 levels do change.
Okay. Fine. We have the next question from another anonymous attendee, and it is, what is the efficiency of ZMYN D8 knockdown in your experiment? Can it be lethal for the cells if knocked out?
Okay, so we have done shRNA based systems for our studies here. So the knockdown efficiency was around 80-85% in our experiments. But you are right. So there are certain issues with generating a knockout context here, because anyways the cells which have D8 levels severely down regulated, even in the knockdown models, we find that the sustenance of the cells are actually compromised. They are slow growing, and there are lots of other problems to the cell. So it's not that it's something that it comes up with a package. You want to study the role of this factor, but it is not that simplistic that that there will be absolute no other effects if you target it in the cells in terms of their expression wise. So it's not lethal. It might not be lethal, but it comes with a lot of other uh, compromised contexts in this particular. If you at all try to knock out them by.
Yeah. In relation to this ZYM and D8, there's one question from Subham. In epigenetic therapy, why you use doxorubicin along with ZYM and D8 but not other drug?
So it's not only doxorubicin. We have done 5-fluorouracil. So as I was mentioning, there are certain standalone drugs which are commercially used in the context of breast cancer. So like doxorubicin, 5-fluorouracil, paclitaxel, like that. We have tried using, and we find a generalized mechanism that when you have this drug and combination with that, the D8, the situations can be saved. So we haven't done our studies in only one drug, but we want to promote that D8 is a factor which can actually function and rescue situations in a generalized manner.
Right. Thank you, Professor Das. So I think we are moving on to the last question by Subham, and it's, what do you think of drug-induced cancer in managing TNBC? Because drug dose and choice of drug is important in killing cancer cells.
Yeah, absolutely true. So what is happening here is that's what we are looking at, and we are observing that in this case the drugs have important effect in killing cancer cells, but that cancer cell killing happens after the drug concentration is elevated beyond certain limit. But if you really if you want to kill the cancer cells, it comes along with several other side effects, which we call it as an off-target effect. So if you really want to play safe, you would like to reduce the dosages of these drugs, and that's how the story has evolved. So, so killing cancer cells by these drugs at a higher concentration is of course a possibility, but it will come along with a baggage of other problems because of the off target. So the question is, how can we combat the situation better? So that's how we are trying to show a dual mechanism where you actually have a chemotherapeutic drug in play, which is much sublethal in amounts, along with playing around with the epigenetic regulator, which can have a better kind of saving the situation in this context.
Okay. So we do not find any question into the Q&A tab. And on behalf of audience, let me thank you, Professor Das, for such an insightful and illustrative talk. I'm sure the audience must have enjoyed it, and this will definitely inspire them to explore the new problems in the discuss area of research. And we hope you enjoyed the Q&A session. Of course, it's different from the in-person events, but still we can't do much.
Yeah.
And
Anyway, I
yeah,
would like to thank you so much, Ajay, and all other people who have helped me to present my work here and share with so many people. And as Ajay mentioned in the beginning of the talk, it is free that anybody who has any other question which was not covered in the QA session and want to contact me personally, feel free. And we would try to discuss and build on our models more and more. And thank you, Ajay, very much for moderating this entire talk and session.
definitely. So, Professor Das, for the participants, we'll send a participation certificate via email. And we would like to thank once again to our participants for their time and wonderful participation. And we'll be back again with another exciting talk. Till then, stay healthy and stay safe. Thank you so much, Professor Das, once again for taking your time and joining us in this today's ACS Science Talk session. Thank you very much.
Thank you.